Good afternoon to everybody. I'm going to speak of an even more rare disease now, but the source of a big frustration for people with me that used to see the classical type of CML, because this is the patient for which you start thinking of being able to tell a positive direction, and indeed you go back to the same survival of CML before imatinib, or even a bit worse than that. So I'll start from the beginning. This is the WHO definition for aCML, so something that looks like CML but does not have any BCR Abelson or any Abelson involvement in terms of genetic alteration. So you know what's the right answer to the question that you received before. And there is also one caveat here, that's the percentage of monocytes that in this classification is given as a percentage, while for CMML is given in absolute numbers, and this generates a bit of overlapping between the two entities, because you can have a patient with more than 1,000 monocytes but less than 10% as in the differential, and therefore in this case the diagnosis is in the limbo of MPN MDS-U. So as I told you, my interest in this disease started from, if you want, clinical frustration, but became, let's say, more interesting, at least from a scientific point of view, when we isolated SETBP1 mutation as being characteristically associated with this disease and present in approximately 1/3 of patients with aCML. And you see this is the paper that we published in 2012. From the list of authors, you can already identify the many collaborators and contributors to my research work, including, of course, Mario, that is sitting here in this meeting. And what we found was that in this disease there is an accumulation of mutation in a very short stretch of 12 amino acid of this gene in what's called SKI homologous region, but does not have much functionality attached to it. But the important thing is that this short stretch of basis identifies a degron, so a recognition point for ubiquitinase enzyme in this protein. And if I can get the light. So what you can see here, and was definitely demonstrated experimentally here, is that if you have alteration at one of these positions, you either destroy the capability of polo-like kinase to phosphorylate residues here, or the capability of ubiquitinase to attach and ubiquitinate the protein. And in fact, as you see here, only the wild-type peptide can bind beta-TRCP, which is the first step in the ubiquitination step, while peptides that are carrying the mutation or that have been dephosphorylated are not able to do that. And indeed, we also confirm this using FLIP3 technology, where you are sure to transfect only one copy of each gene. And again, you can see that the amount of protein is dramatically different if you have transfected one copy of the wild type SETBP1 of one copy of the mutated gene. So in other words, the net effect of this mutation is to decrease the degradation of the protein and to increase the protein level of the gene. I'll go back. From a clinical point of view, what we saw, and has been confirmed by papers from both ILF Teferi group and also from Professor Solari group, is that the presence of mutation of SETBP1 is associated in a multivariate analysis with a definitely worst prognosis. And if you see here, in fact, if you see here, the median survival goes from more than 40 months here to approximately 20 months for ZBP1 mutated patients. So we wanted also to proceed in the knowledge of what ZBP1 does in this case. Everybody knows that ZBP1 binds SET, that the name comes from this, and that the binding of SET then stabilize SET and lead to the inhibition of PP2A. But this is one very known way for ZBP1 to work. But we also know, for example, one thing, that while PP2A is distributed both in the nucleus and in the cytoplasm, ZBP1 is completely nuclear. And if we do also cytoplasmic and nuclear extract, we confirm that most of ZBP1 is either wild type or mutated, is present in the nucleus and very little in the cytoplasm. So we thought that there should be something in addition to this SET binding activity of ZBP1. And to investigate this point, we moved to ChIP-seq experiment, where we precipitate using an anti-ZBP1, or let's say an anti-TEG antibody, and then we run a ChIP-seq analysis of all the DNA that is precipitated by the antibody. By doing that, we can see accumulation of sequences in a certain part of one gene. These are very preliminary, but by doing this, we identified a number of genes where ZBP1 is binding. I will show you just one example. This is one gene. You see the ATG here, and these are the number of sequences that are mapped to the various regions. As you can see, there is quite a large number of signals in the promoter region of these genes. So to couple this NGS data with functional data, we used RNA-seq to build gene expression profiles from ZBP1 positive or negative cells. And what we can see is that if you look at the entire number of the genes, you see that there are many more genes that are upregulated when they bind the promoter, when ZBP1 bind their promoter, than downregulated. It's only one gene that looks to be downregulated and show binding of ZBP1. Of course, there is also a large number of genes that show binding, but without having an evident effect on transcription. We used here a cutoff of 2 times to consider to be modified by the binding. But the main message is that the binding of ZBP1, we saw it's definitely linked to upregulation of the genes that where the binding happens, and we also have confirmation of this by real-time PCR. There have been also data from other groups, in particular from Dr. Maciejewski, of other genes being regulated or at least associated with the presence of ZBP1. They mostly noted RUNX1 as one gene that is, of course, interested for hematological diseases. I have to make the caveat that they always use the murine genes instead of the human one. We tried also to see whether RUNX1 was really down-regulated in human cells. Indeed, it is down-regulated, but if we look at the binding, we find a very weak binding, and if we try to ascertain the direct effect of this binding is weakly positive. So the downregulation that has been observed and we confirmed here is probably not due to the binding of ZBP1 to the genes, but to some more indirect effect. Now I'll change type of gene, because we also identified a second genes that is involved in the pathogenesis of aCML. And this is ETNK1. ETNK1 again show in approximately 10% of patients a mutation in one or two positions, so very conserved and very focused type of mutation. And ETNK is an enzyme that is involved in the synthesis of phosphatidyletanolamine, which then can go to a membrane lipid and anchoring other proteins, or being involved in cell motility. So we try to understand what the mutation does. We have much less information than for ZBP1, but what we can say is that if we look at the level of the result of the enzymatic reaction, which is phosphoetanolamine, and we compare this with phosphocholine that is in the parallel pathway, we can see that cells from patient with mutation compared with patient with no mutation, or compared with normal cells from the same patient, show a reduced level of phosphoetanolamine, suggesting, let's say at least, that this mutation could decrease the normal function of the enzyme. We cannot say whether this is just a loss of function mutation or a shift of function mutation at present. In aCML, there have been also a third gene that has been described recently as being mutated, which is the receptor for CSF, and it's been described in two papers that I'm showing here. These two papers, however, have not really looked in specific to aCML, because they look to both atypical CML and neutrophilic leukemia. We just looked at our own series of 70 aCML patients, and we found that mutation of this gene was present in 5 out of 70 patients with 7%, and the 95 confidence interval is between 3 and 15% of them, so in a quite low proportion. As Professor Solari pointed before, of course, these mutations do not come as isolated mutation. Even in the original publication, we could see that there was a tendency toward ZBP1, for example, to associate with ACXL1 and to be mutually exclusive with TET2 mutation. So we reverted to clonal analysis of this patient, having both exome and RNA-seq on one side, and doing targeted resequencing of growing colonies. In this way, we can reconstruct the clonal architecture of this patient, and we find definitely a huge heterogeneity in this patient. So, for example, in this case, we had these 4 mutations. We had both ZBP1, ETNK, ACXL1, and CBL. And by looking at the clonal architecture, we can definitely see that, at least in this patient, it's not general, but I would say it's prevalent. ZBP1 was definitely a late mutation. CBL was more intermediate, as ACXL1, while ATNK was an early lesion. In all these steps, as you can see here, there is an animation missing here. in all these cases, we can definitely see that at each level there is a tendency for a CBL heterozygous mutation to become homozygous that you can see here. And we saw this in other cases as well. So these are 3 cases with different evolution, but in all these cases you see that at least in these 3 cases ET and K tend to be an early event, while SETBP1 tend to become later on. And also we always see this shift toward developing homozygosity for CBL mutations, as we could also see using CGH analysis as being linked to a somatic uniparental disomy for the region where CBL is present. So I will finish here my talk with one proposal and one hope. The proposal is for the people involved in the classification of this disease to try to sort out the problem of overlapping between CML and aCML in terms of absolute versus relative number of monocytes. This would, let's say, help the research in clarifying who is who. And in one hope, of course, the hope is that this was the survival of, not of aCML, but was of CML before imatinib, to be able at the end of our exercise to transform it at least a little bit as it is CML today. And I will close here and will thank all the people in the lab that helped me in reaching these results. Thank you.